fertilization in that between the. So the Mendel experimental design could use the self-fertilize and produce the to true breeding generation. So after the second stage means produce first fertile generation means F1 generation, that is transfer pollen from one parent to another, that is the pollen transferred from white flower to carpel of purple flower. So here the anthers are removed, so which is encourage the cross pollination. And the third stage left F1 generation self fertile means self fertilize to produce a second filial generation means F2 generation. So all the purple flowers F2 generation to produce would be all the purple flowers. So here the result of Mendel's process means that flower color means the purple and white, seed color yellow and green, seed shape round and wrinkle, pod color green and yellow. So is the what F2 generation, that is the dominant and recessive character here 3 is to 15 is to 1, and seed color here 3 is to 1 is to 1, and seed shape that is the ratio of dominant and recessive character means phenotype and genotype ratio 2 is to 96.1. These are there. And so genetic terms, what are the genetic terms? We have to use it here, genotype and phenotype. What is genotype? What is phenotype? Here we have to discuss that one. The genetic makeup of an organism are alleles carried by individual or arrangement of genes that produces the phenotype, that is the genotype. So what is phenotype here? Physical characters or appearance of individual, that one we call as a phenotype. So capital T here, that is a tall one, that is a homozygous dominant. So tall pea plant, that is the capital T and small t. This one also tall plant. It is the heterozygous plant. And small t, this one is the dark plant, which is the homozygous recessive character is present. So here, see here that I show to read. See here, the homozygous parent can only pass one form of an allele to their offspring. Only one character is passed to the uh, that one, that their offspring means the daughter cells. So homozygous parent and gametes are there, and heterozygous parent can pass either two forms of allele to their offspring, that is the heterozygous parent and gametes. Dominant alleles and recessive alleles. What are the dominant alleles? What are the recessive alleles? Here, the general terms for an allele that makes the presence of another allele in the phenotype, that is the dominant allele. What is the recessive allele? In the general term, an allele that is masked in the phenotype by the presence of another allele, that is the recessive, means which one is the inside there. Yeah. So a distinct 1 is to 2 is to 1 ratio Uh, 1 is to 2 is to 1 ratio means in the Mendel went to an experiment here F2 plants foster traits on the subsequent generation. Mendel found that the recessive 1 by 4 were always the true breeding. This result suggested that for the entire sample, here 3 is to 1 ratio that Mendel observed in the F2 generation. So it is designated as 2 is to 1 is to 2 is to 1 ratio. Means here the purple breeding dominant individual here one is and two not two breeding dominant individual and 1 by 4 here three breeding recessive individuals are there. So here see that in the figure. I show to the figure here. Wait. So see here this figure here what shown here the parent generation is the purple and white flower which is the F1 generation in the self fertilized and F2 generation to produce the 1 is to 3 is to 1 means that is the here purple and white color. Okay. The Mendel of heredity for his experiment, the Mendel was able to understand four things about the nature of heredity. So first one, the plants he crossed did not produce progeny of appearance. Intermediate appearance. The second one, the Mendel is learned that for each pair of alternate forms of character, one alternate was not expressed in the F1 hybrids, although it reappeared in same F2 individuals. Okay. Mm. The third stage is that is the pairs of alternate traits is examined, segregated among the progeny of a particular cross, some individuals exhibiting one trait, some of other. And the fourth one, these alternative traits were expressed in the F2 generation in the ratio of 3 by 4, dominant to 1 by 4 recessive. So here, Punnett square board. So his Punnett board square board also is introduced. See here. So I'll show to this one. Hmm. So this one is the Punnett square board here. So a Punnett square is used to show the possible combination of gametes. So segregation and fertilization as chance events. In this white flower and purple flower, the gametes we are to place in this one. That is the capital P, and small p. We are to F1 gametes are there. So the second failed generation means F2 generation. And purple flower here, purple flower here. One is the dominant and recessive character. Both characters are there. So here in the F2 generation, the phenotype ratio here 3 is to 1 and genotype ratio here 2 is to 1 is to 2 is to 1. That is the general Mendel is the phenotype ratio and phenotype ratio. To explain these results, Mendel proposed a simple model. It has become one of the most famous model in the history of science, that is containing simple assumptions and making the clear predictions. What is that one? The Mendel has five elements. That is, the parents do not transmit physiological traits directly to their offspring. Rather, they are transmitted discrete information about the traits, what Mendel called factors. So each individual two factors that may code for some of trait to alternative traits characters are there. Not all copies of factor are identical. The two alleles, one contributed by the male gamete and another by female, do not influence each other in any way. The fifth one, the presence of a particular allele does not ensure that the trait ended by it will be expressed in an individual carrying that allele. So Mendel's first law of heredity, that is the segregation, means the individual segregation are there. The alternative allele, okay, that is here. Let's so, so Mendel's first law of heredity, that is the segregation, means the alternative allele of character is segregated from each other in heterozygous individuals and remain which one is the distinct. There is the phenotype versus genotype. Capital S and capital S small s, two different genotypes, the same phenotype is present. So they have the here small and capital. So they have the same phenotype, but the different genotype is there. How do they distinguish these two genotypes is experimentally in the. So how can we know it is the homozygous or heterozygous? To introduce only the test cross only, a maintain between an individual of unknown phenotype and a homozygous recessive individual. Here, the test class confirmation of the segregation only, through the segregation only. Here we have to confirm that one is a homozygous or either heterozygous. A homozygous recessive, that is the white flowers are there, and another one is the dominant phenotype, that is the all offsprings purple, therefore unknown flowers was homozygous. Here half of offsprings white, therefore unknown flowers were heterozygous. To discuss that one, the law, Mendel's second law, that is the independent assessment. The independent assessment here, that F2 generation, the phenotype and the Mendelian inheritance is not always easy to analyze. In these decades, the following of um, rediscovery of Mendel, many investigators set up to test Mendel ideas. However, in the attempting of confirm Mendel theory, often had trouble to obtain the same simple ratio he had the reported. Most of the phenotypes reflect the action of many genes that act subsequently or jointly, and the phenotype can be affected by alleles that lack the complete dominance and the environment. So here another so here pleiotropic effects. What are the pleiotropic effects? Is often an individual allele will have more than one effect on the phenotype. Such allele is called as a pleiotropic. When the French genetist, that is the Cunard, studied yellow for mice, he was unable to obtain the true breeding yellow strain by crossing individual yellow mice with each other. So, in individual homozygous for the yellow allele died because the yellow allele was pleiotropic. So a pleiotropic allele may be dominated with respect to one phenotypic consequence and recessive with respect to another. The pleiotropic effect or characteristic of many inherited disorders, sickle cell anemia. These disorders, multiple symptoms can be traced back to the single gene defect. In the sickle cell anemia is defected in the oxygen carrying hemoglobin molecule cause the anemia, heart failure, kidney failure, and enlargement of the spleen, and many other symptoms are there. So another one is the lack of the complete dominance. So the not all the alternative alleles are fully dominated or fully recessive in heterozygotes. Some pairs of alleles instead produce a heterozygous phenotype that is either intermediate between those of the parents or representative of both parental phenotype. For example, in the cross of red and white flowering Japanese pole cloud for F1 offspring had pink flower, indicating that neither red or white flower was dominated. Environmental effect, the degree to which all the alleles expressed may depend on the environment. Some alleles are heat sensitive. For example, the traits influenced by such as alleles are more sensitive to temperature or light than other producers of other alleles. Okay. The Arctic foxes make four pigments only when they are whether in the warm condition. So that is the environmental effect also there. So another one is the another aspect is epistatis. When the individual heterozygous for two different genes made for different phenotypes are possible among the progeny. So offspring may display the dominant phenotype for both genes, either one of the genes or for neither gene. So sometimes, however, it is not possible for an investigator to identify successfully each of the four phenotype classes because two or more of these classes look alike. Such situations is provided confusing the investigator with the following Mendel. So here what's happening? The Mendel had predicted 16 equally possible ways gametes could be combined. So, how many of these were in each of the two types of immersion obtained? Is multiplied the fraction that were the pigmented produced by 16 to obtain 9 and multiplied the fraction that were not by 16 to obtain 7. So the immersion had modified ratio of 9 is to 7 instead of the usual 9 is to 3 is to 3 is to 1 ratio. Okay. Why the immersion ratio is modified? When the genes act sequentially, as in by chemical paths, an allele is expressed as a defective enzyme early in the pathway blocks the flow of material through the rest of the pathway. Such genes interaction where one gene can interfere with the expression of another gene is the basis of the phenomenon is called epistasis. The pigment anthocyanin is the product of two steps by chemical pathway, the staining molecule, the intermediate and anthocyanin. So another multiple alleles of ABO blood group, that is the a gene may have more than two alleles in a purple in a population, and most genes possess several different alleles. So often no single allele is dominant. Instead, each allele has its own effect, and the alleles are considered as codominant. This gene is encoded as enzyme that adds the sugar molecule to lipids on the surface of red blood cells. The genes that encoded the enzyme is designated capital I has three common alleles, that is capital I, capital B. Those product as is the galactose and capital IA. Those product is galactosamine and which is the that is the so the next one is the incomplete dominance here. Incomplete dominance. Red and white flower, that is the gametes F1 generation means the pink color is appear. Okay. So another topic is linkage. So I show that linkage means the discovery of linkage. The principle of linkage was introduced by the Betson and Panett in 1906 in Street P. So that is, it was put forward as a regular concept by the Morgan in 1910 from the his work in Drosophila melanogaster. So see here, what is the linkage? So meaning of linkage, see here, I show that figure. So linkage, see here, that is the meaning of linkage. The linkage is the phenomenon of certain genes stating together during the inheritance through several generations without any change or separation due to their presence on same chromosome. So that one is the linkage. What is the characteristic of linkage? The linkage involves two or more genes which are linked into some chromosome in a linear fashion. The linkage reduces variability. It may involve either dominant or recessive alleles, or sometimes some dominant and some recessive alleles. It usually involves those genes which are located close to each other. Um, it is the strength of linkage depend on the distance between the linkage genes. Lesser distance, high the strength of linkage. So another one is the gene linkage. Mm, genes in linkage here, linked genes. These genes do not show the independent assessment. It occur in the some same chromosomes. And another unlinked genes. These genes shows the independent assessment. Some theories proposed by these batsmen and Morgan that the linkage theories are there. What are the linkage theories? Here is the differential multiplication theory, and another one is the chromosomal theory. These chromosomal theories proposed by the Morgan and the differential multiplication theory was proposed by the Batson. So, what is the Batson theory here, which is introduced by the 1930? The Batson he said that after the segregation of genes during the gametogenesis, certain genes multiply faster than other. This one. So, and another one is the chromosome theory, which is proposed by the Morgan. So, these genes which shows the linkage are located in the same chromosome. The distance between linkage genes in the chromosome determine the strength of linkage. The genes lies in the linear manner in the same chromosome. Some another topic is some kinds of linkages are there. On the basis of crossing over, on the basis of chromosome involved, and on the basis of gene involved. So these three types of linkages are occurred. In the base of crossing over, here the complete linkage, the genes are located in the same chromosome or inherited together over the generation due to the absence of crossing over. Okay. It is the rare but has been reported in the male Drosophila melanogaster. The incomplete linkage. Okay. And this one, the genes present on the same chromosome have a tendency to separate due to the crossing over. They produce the recombinant progeny beside the parental type. So here on the basis of chromosomal involved, based on the chromosome involved, the location of genes on the chromosome linkage is categorized into autosomal linkage and allosomal linkage. In the autosomal linkage, it is refers to the linkage of those gametes which are located in the autosomes, means other than the sex chromosomes, that is the autosomes, and allosomes linkage refers to linkage of genes which are located in the sex chromosomes, that is X or Y. Okay. So on the basis of gene involved, that is the depending on the whether all the dominant or same dominant or in the recessive alleles are located together, a linkage can be categorized into two types, that is the coupling and repulsion phases are there. Coupling phase and repulsion phases are there. In the coupling phase, the dominant allele and the recessive allele is present on the same chromosome. Some coupling phases are there. And repulsion phase, the dominant alleles of same genes are linked with the recessive alleles of other genes on the same chromosome. That is the repulsion phase. Here, the linkage group. Another term, the linkage group is the linearly arranged group of linkage genes which are normally inherited together. That is the linkage gene. Example, Drosophila melanogaster have four linkage groups are there. In human beings, 32 linkage groups parents are there, means 23 pair groups are there. So what is the significance of linkage? It is reduced to chances of formation of new combination of genes in the gametes. It helps to keeping the parental and other specific sites together. The linkage plays an important role in the determine the nature and scope of the hybridization. Another topic is also another topic is the crossing over, which is crossing over is the what is that here? I show that on diagram. Yeah. So what is the crossing over? The crossing over is the exchange of genetic material between the homologous chromosomes that results in the recombinant chromosomes. Between the homologous chromosomes, the exchange the genetic material. So, what are the characteristic feature of the crossing over? Here, the crossing over occurs between the non-sister chromatids One chromatid from each of the two homologous chromosomes is involved in the crossing over. The crossing over leads to recombination or a new combination between the linkage. The crossing over generally leads to the recombinant type, recombinant type or crossing over types, and which are two parental types or non-crossing over types. So generally, which is leads the exchange the equal segments, or sometimes genes and recombination is always reciprocal genes are there. So stage at which is the crossing over occur in the meiotic crossing over takes place during the parent packaging stage of the prophase of meiosis one. The packaging stage only the crossing over is occur. The packaging stage is also known as the recombinant stage. The crossing over occurs when the homologous chromosomes are in the four chromatid or tetrad stage occur in the packaging stage. So here some types of the crossing over. Mainly here two types of crossing overs are identified. Here somatic or mitotic crossing over, and another one is the germinal or meiotic crossing over. It is and meiotic crossing over another uh, division divided into two types are there: equal crossing over and unequal crossing over. So somatic crossing over, either mitotic crossing over. This one, the crossing over occurs in the somatic cells during the mitosis stage. It is rare and has no genetic significance in the somatic crossing over. Another type is the meiotic crossing over. This type of crossing over takes place in the germinal cells during the meiosis that produced the gamete. It is universal and has a great genetic significance. Again, here germinal crossing over can divided into two types are there: equal crossing over and another one is unequal crossing over. In the equal crossing over, the segments exchange between the chromosomes are of equal in size. It is divided into three types according to the number of points at which is the occur in the single cross, double cross, and another one is the multiple crossing over. So single crossing over, already I showed to the picture. Yes, see here. So see these diagrams for the single and double multiple crossing over diagrams already shown. See here. Oh, and single crossing over. This one, the chromatids break and unite at a one point only. At the single point only, there are the reunit or breakdown at a single point. That one is the single crossing over. And the double crossing over. During this type of crossing over, chromatids break into reunit at 2 points in the same tetrad state. And another one is the multiple crossing over. In the multiple crossing over, the chromatids break and reunite at many points in the tetrad. So that's one here multiple crossing over. Another type of crossing over in the meiotic crossing over, that is the unequal crossing over. The segments exchange between the chromatids are unequally, so that one chromosome receives an extra gene and another gets one geneless. So that is the unequal genes are there in the tetrad. So next one is the mechanism of crossing over. Here some aspects are involved in these crossing over synapses and the tetrad formation, exchange of the chromatic segments and terminalization. These steps are there. What is the synapses here? In the zygotic, during the prophase one, the homologous chromosomes of each pair come together and line up to side by side. This pairs of homologous chromosomes is called as a synapse. So here, what happens here? The homologous chromosomes, so they become a pairing in the prophase phase. So another one is the tetrad formation. Another stage is the tetrad formation. Here, the two chromatids of chromosomes are referred as a dyad. A group of four homologous chromosomes so, that have two synapsid homologous chromosomes that is known as tetrad. The two chromatids of same chromosomes are called sister chromatids. These two chromatids, one of the chromosome and another self homologo, that one is called as a non-sister chromatid. Hmm. So another one is exchange of chromatid segments. Here, two non-sister chromatids tetrad break at equivalent locations. Here, the break and transpose and join their receptive breaking ends of opposite chromatids. This completes the process of crossing over. And unexchanged chromatids are called as a parental or non-crossing over. The exchanged chromatids are called as a recombinant. So the recombinants, so you see here, so that's the diagram. So see here, this is the um, crossing over process are there. This one is a non-sister means chromatids and sister chromatids are homologous chromosomes and non-sister chromosomes. They are exchange their DNA between the means non-sister chromatids. So reunion of another one is the terminalization. Last stage is the terminalization. Here, the completion of crossing over that end of the taxing stage, beginning the diplotin stage. The synaptic forces end and homologous chromosomes are separated. At the point at which this separation does not occur is called as a chiasma. Mm, the chiasma means chromatids is separated progressively from centromere towards the chiasma, moves like a zipper towards the end of the tetrad. Mm, the sinking of chiasma towards the end of the bivalence called as a terminalization. Sinking of chiasma towards the end of the bivalence are called as terminalization. See here the terminalization is shown. So So termination process is there. This one is the terminalization. Okay. See here the terminalization. The homologous chromosomes and bivalent tetras. Between the bivalent tetras, the genetic material is not exchanged, that we call as a chiasma. So effect means the factors affecting the crossing over, that is the material age of effect and the temperature, nutrition, chemical effect. Another effect is the chromosome effect, centromere effect, mutation effect, and another one is sex determination. What is the significance of crossing over? So the crossing over has helped in establishing the concept of linear arrangements of genes. The frequency of crossing over helps in the mapping of chromosomes, that is, the determine the location of genes on the chromosomes. It is an important factor in the sexual reproduction. These crossing overs also increase the variation, which is vital for evaluation, and it is helped in the plant breeding also. And difference between the linkage and the crossing over, it gave some differentiation between the linkage and crossing over is there. It keeps the genes together in the linkage, but the crossing over leads to separation of linked genes in the linkage, which is involved individual chromosomes. So the crossing over involves exchange the segments between the non-sister chromatids of homologous. In linkage, the number of linkage groups can never be more than applied chromosome number. The crossing over frequency crossing over can exceed 50%. It is reduces variability. In the crossing over, it is increased the variability by the formation of new recombination. So this is the topic about the linkage and crossing over. Okay, thank you for attending the class. The tomorrow next class we are to discuss another topic. So thank you everybody for attending the class. Thank you. Thank you, one and all.